First, I'm happy to be here, even if it's via Zoom, which is certainly not my weapon of choice, but I'm honored to have been asked, and I'm glad that you guys sort of pulled this off at this scale, which I know was sort of an unprecedented feat for Ashley and Christina and everybody else involved in the background of this. So let me start by thanking you. And as sort of a brief overview, I guess I'm a little embarrassed to admit sort of how cheesy this is, but my research is really driven by just a childlike fascination with the brain, especially the sheer power and scope of biological computation. And the craziest part of this to me over the last few years of thinking about it is that, generally speaking, I take this phenomenon completely for granted. The fact that day in and day out, you can identify milk in your refrigerator, tie your shoes, walk to the lab, all of which points to a truly incredible robustness and reliability in a vastly complex system. The work in my lab is directly concerned with how neural computation achieves this robustness and what that means at the level of neural dynamics. Here today, I'd like to introduce a line of work from my lab that we published last year that directly tackles the problem of stability in complex systems and leads us to ongoing work that leans very heavily on cutting-edge detailed models, which are kind of the theme of the day here. Let's see if I can control the screen. There we go. Great. Now, questions of stability in the context of neurons have been raised since at least the 1950s. The broader idea of self-stabilizing biological systems was crystallized in the 1930s by Walter Cannon. Cannon introduced the term homeostasis, which refers to the ability of a system to maintain some set point despite fluctuating conditions. Think, for example, about how a thermostat, a furnace, and an air conditioner can keep your house at the same temperature throughout the year. Now, there's been rigorous work to identify a host of homeostatic mechanisms in neurons, mostly at the level of synaptic weights, cell membranes, and single neuron firing rates. However, brain function and neural computation emerge from the ability of neurons to influence one another's activity across networks and over time. Which brings us to the concept of the computational regime. Given the many often non-linear forces that influence a neuron's function, it's hard, for me at least, to imagine how a network generates reproducible behavior under even slightly variable conditions. And yet, day in and day out, I can still find my way home and recognize my family. So despite this challenge, neural computation tends to work, such that activity in one neuron, say at the top right of the screen, has a fighting, or the bottom left of the screen, has a fighting chance of influencing activity in another neuron, say at the top right. And for this to be true, there must be higher level constraints on the system, perhaps at the level of, say, connectivity, topology, and excitability. And here, the term computational regime describes the collective impact of a full set of constraints that govern the interaction of neurons in an ensemble, and ultimately govern the kinds of computations that can be performed. And key features of network computation are tethered to this computational regime. First, the dynamic range of a network. That is, the difference between the weakest and the strongest stimuli that a network can effectively respond to. Second, the ability of neurons to share information at any distance across the network. Similarly, the ability of neurons to share and maintain information over time. And finally, a network's regime limits the number of patterns that can be observed, both in space and in time. And together, these features determine the computational capacity of a circuit or a network. And with this understanding, it seems important to me to ask, what is the computational regime of the cortex? How is it implemented? And how stable is it? Or how is it stable? So I think that a concept from physics is relevant here, so just bear with me for a moment. And this is the concept of a system poised at a phase transition. As a brief example, consider a magnet. The interaction of iron molecules is very, very strong. That's why it's a magnet, such that neighboring molecules force each other to align in the same direction. If you were to flip one molecule, it would be quickly corrected by its neighbors. In contrast, if you heat a magnet, the interactions fall apart and molecules orient themselves randomly. In this case, the effect of flipping a molecule is immediately swallowed by noise. However, the magnet exhibits some very peculiar and desirable properties at the precise point between highly structured and chaotic regimes. At this point, this phase transition is called criticality. And at criticality, there's a swirling mix of order and disorder. But perhaps most importantly, at criticality, flipping a single molecule has the highest chance of producing a detectable change across any distance within the magnet or over any amount of time. And what that means is that criticality simultaneously maximizes dynamic range, information transmission, and information storage. And crucially, it's the only known computational regime that does this. Which leads us to the brain. If you think about neural systems, there's also a phase transition between a regime dominated by inhibition, where there's limited activity, and an overexcited regime, where there's runaway, perhaps epileptiform activity. And like the magnet, at the phase transition, long-range structured patterns can emerge, and at this transition, all of the computational features that I listed a moment ago are simultaneously maximized. So let me show you a simple example. Here is a toy model of interacting bits that, with some certain probability, will excite their neighbors. On the left is a subcritical system. This one is defined by a very weak interaction magnitude. The parts don't have very much influence over one another. Activity in this system, blue in this case, will have trouble propagating. On the right is a supercritical system. Activity spreads, oh, I want to click. Supercritical, go. There we go. Activity spreads far too rapidly in this system. And in either case, it would be extremely difficult to move information across the network, across this field. And in the middle of these two examples, though, is a critical system. See if I can get that to play. No, hold on a sec, bear with me. Restart these three. Ashley, do you also have control over the screen here? Oh, I can do that. Great. So here are the three systems operating, and the central system, again, is critical. And the intermediate interaction regime that defines that central system gives rise to events, or bursts of blue here, that span all temporal and spatial scales without ever blowing up or decaying. So given that you are an audience of people interested in computational modeling, this is all probably very exciting, but quite abstract nonetheless. And lucky for us, measuring criticality in the brain takes advantage of the same principles. Consider the plot on the left. This is a raster plot that shows the spike times of 5 neurons recorded, in this case, in the visual cortex. And notice that, much like the spreading blue events in the prior example, spiking tends to emerge in spatiotemporally clustered bursts, which have been whimsically termed avalanches. I've outlined the avalanches here in green. This is even more obvious if you look on the right, where I've collapsed the spike times into a single vector. Avalanches can then be easily measured in terms of size, which is the number of contributing spikes, and duration. Now, recall that critical systems, by definition, generate events of all sizes and are thus scale-free. Apropos this point, we can then plot the probability of observing avalanches of a given size, shown here in yellow, or a given duration, shown in blue. And also take note that these are log-log plots and that these distributions are well fit by a line. In other words, they obey a power law. And a power law has the special property that it doesn't change when you zoom in or zoom out. It is the mathematical definition of being scale-free. Now, for a system to be truly critical, those two measurements, size and duration, must scale together according to a third power law called the scaling relation. And the predicted scaling relation is derived from the size and duration exponents, tau and alpha here, which can then be compared to the observed scaling relation. Here, the predicted fit is shown in solid gray, while the observed data are plotted in purple and fit with the dashed line. We took advantage of this and produced a novel measure of how close a system sits to criticality. We simply quantified the deviation between the dashed and the solid lines, and we call this DCC, the deviation from criticality coefficient. So if that slide was a lot for you to digest, fine, same here. But the takeaway point is that we developed a single scalar measure of how close to criticality a system is, and it works like a golf score. Lower DCC is better, and a DCC of 0 indicates a perfectly critical system. All right, with that out of the way, to validate DCC, we took advantage of a simple deterministic model in which we could control the ground truth with regards to criticality. And for the aficionados in the audience, if the maximum eigenvalue of the adjacency matrix is exactly 1, the system will exhibit critical dynamics. So here's a plot of the avalanche size distribution from a critical model and the corresponding DCC. In contrast, here are the distributions and DCCs from supercritical and subcritical models. And when we run many simple models, you can easily see a U-shaped curve whose minimum sits at the critical point. And based on this curve, we suggest that a DCC value of less than 0.2 is consistent with a near critical regime. And turning to real data, we checked the DCC and size distributions in a few previous recordings from the lab. And here I'm showing you avalanche distributions from the visual cortex of 3 separate animals, each of which reveals a near critical DCC. All right, with all of this in hand, we wanted to then ask that core question that I raised at the top, whether criticality is a set point for emergent dynamics in the cortex of freely behaving animals. However, studying homeostatic principles in the brain is a tricky task for 3 reasons. So let's briefly return to that analogy of temperature in your house. First, you would need to demonstrate that the temperature is stable at baseline across a decently long period of time. Second, you need to figure out how to significantly perturb or challenge your system, perhaps by opening a bunch of windows in February. And finally, you would need to be able to maintain that perturbation and your observation long enough to convincingly demonstrate whether or not your furnace can mount an appropriate defense. The most widely used homeostatic challenge in neuroscience is that of monocular deprivation. This capitalizes on the fact that the rodent visual system is highly lateralized. Closing the left eye deprives the contralateral visual cortex of organized light input, but it leaves, so there you can see the deprived eye and the deprived hemisphere, but it leaves the right visual field and thus the left hemisphere almost entirely unaffected. In this context, we implanted microelectrode arrays bilaterally into freely behaving juvenile rats and proceeded to continuously record neuronal spiking for 7 days. After 24 hours of baseline activity, we briefly anesthetized animals and we closed one eye. And we've shown that previously that this manipulation drives a strong homeostatic response at the level of neuronal firing rates. And that effect, that homeostatic effect at the level of firing rates, is obvious in this data set. So here I'm plotting the mean normalized firing rate of excitatory cells in both hemispheres. On the left is the deprived hemisphere. Gray and white bars represent 12 hours of dark and light respectively, and the timing of monocular deprivation is marked at the top. And as you can clearly see, the neuronal firing rates drop significantly in the early phase of deprivation, and then despite the fact that the eye is still closed, firing rates return to baseline levels. And the control hemisphere on the right shows no such drop and rebound. So herein we have a data set in which we can demonstrate a homeostatic challenge and a response. And it is in these data that we then asked questions of criticality using our measure DCC. The first noteworthy point is that, lo and behold, the visual cortex was robustly near criticality for the entire baseline day, despite massive changes and rapid fluctuations in behavior and things like transitions between light and dark. So then the key question became whether or not criticality is disruptible. If not, that would suggest that criticality is perhaps an inevitable epiphenomenon that arises from just connected neurons. And this would immediately render it totally uninteresting. And if criticality is disruptible by monocular deprivation, what is its time course? Is it simply a computationally expensive readout of cell-level changes in firing rates? This would be mildly interesting, I guess, but largely suggests that emergent properties are entirely the result of cell-level control over spiking rates. So, given that spiking dropped by more than 60% in the top left plot, this was my prediction. It seems kind of obvious to me. Of course, the computational regime is going to fall apart if you rip out more than half of the spikes. But as usual, I was impulsive and wrong. DCC more than tripled in the first 4 hours of light exposure during deprivation. This preceded a drop in firing rate by more than 30 hours. And then criticality was precisely restored at the nadir of firing rate suppression, again, more than 30 hours ahead of the firing rate rebound, that main homeostatic effect that we've been measuring. And this whole time, DCC in the control hemisphere was stable across the entirety of the recordings. So I assert or suggest that these data reveal an actively maintained set point for cortical dynamics that A, is predicted by theory, and B, has explanatory power for circuit function, and C, is firing rate independent. It can't be explained by local cell level changes, at least in the cells we're measuring. Now, because emergent phenomena are quite difficult to untangle in intact biological systems, we turned again to our simple model with hopes of understanding what aspects of the network might be most influential in asserting criticality. Our model on the left had inhibitory and excitatory neurons and constant excitatory input. And we simulated monocular deprivation by drastically reducing that input midway into the experiment. Now, our goal was to search a very large parameter space and ask which models successfully recapitulated our experimental data. And to be considered successful, models had to first exhibit firing rate homeostasis, a drop and a rebound. Second, models had to have baseline criticality and homeostasis therein. And then third, models had to exhibit an inhibitory firing rate drop before excitatory, which is a finding we published previously, actually in 2013. So we explored hundreds and hundreds of models, and yet only 3 discrete combinations of parameters were sufficient to reproduce our results. These important factors all centered on inhibition. The solution space here in the bottom left of the plot is defined by the inhibitory to excitatory connectivity, the number of inhibitory neurons in the network, and the proportion of inhibitory neurons that receive external drive. So to further this, we next tried to test the ability of non-inhibitory parameters to influence criticality. And we took one of the failure models near a successful one, here the little white tile outlined in dashed red, and we tried to rescue that model by then adjusting many combinations of non-inhibitory parameters. Very quickly, those were homeostatic plasticity, Hebbian plasticity, and excitatory connectivity. Now, some of these models, nested within that red tile, rescued the offset in the excitatory and inhibitory suppression. Some of the models revealed firing rate homeostasis, and some of the models showed critical dynamics at baseline. But none of these models successfully captured all of the features together. So we take these results as strong implication that inhibition is crucial for asserting the computational regime of the network. So let me stop there with data and distill 4 key points that I've covered. First, simply that the concept of the computational regime is powerful and worth careful study. Second, that criticality is an ideal regime, consistent with much of what cortical circuits do on a daily basis. Third, I showed you that the cortex is robustly near criticality. And finally, that criticality is consistent with a homeostatic set point for cortical dynamics. But to really leap from theory and basic observation into biological application, we will need to address one obvious question, which is, what are the circuit mechanisms of criticality? And I've thought a lot about this question, and our path towards its answer is largely, I believe, why I was invited to speak here today. Thank you, Anton. So, complex brain function emerges from the interactions of a vast array of cellular and subcellular components. This is clear from everything we've heard this morning. And systematically probing the combinatorial explosion of these at the bench is a recipe for insanity and a career of failure. But that doesn't mean we don't need to fundamentally move in that direction. And this is where, from my perspective, better and more detailed models become essential tools for progress. Consider the simple model. Consider the simple model that informed a lot of the results I shared with you. Using this unstructured network with only 2 cell types, we were able to compellingly point to inhibitory terms as a great place to start. However, there are many species of inhibitory neurons, many anatomically specific populations, and a staggering range of variables that affect their performance. Essentially, we're still lost at sea. Thankfully, though, there are now open source, unprecedentedly detailed, biophysically realistic models available to all of us. Oh, no. There we are, you should recognize that. For example, Anton here at the Allen and his team produced an exquisite model of the visual cortex with 17 cell types, thalamic input, and 230,000 individual neurons. And this model does a very good job of recapitulating cortical responses to visual stimuli. And my lab is now using this model to identify plausible circuit mechanisms for emergent dynamics, such as and including criticality. So let me just wrap up by giving you a preliminary hint of what this looks like. Here's a raster plot of 50,000 neurons in the model's core. Each tiny point represents a single spike. Cells are organized by layer from top to bottom, and the color broadly indicates cell type. The visual stimulus is indicated by the black line at the bottom. It turns on at 500 milliseconds. And the advantage of this type of model is massive. It gives us the ability to make specific and nuanced manipulations that correspond to biological details, and then carefully measure their impacts on complex network properties, such as criticality. Some of these manipulations simply destabilize the network. Here's an example. When I click, I'm going to delete layer 4 PV interneurons. That's the second pink band from the top. And when I do this, you will see that the entire network shifts towards saturation. I'm going to click in, so watch closely. It's not super obvious. I'm going to click in 3, 2, 1. There. So you can see that that second pink band disappeared, and there's a lot more blue in that network. And while that clearly changed the model.
Keith, it's 10 minutes.
Okay, great. I'm my last slide here. Thank you. The impact of this deletion is obvious in first order statistics. So here's a plot of the firing rate by cell type at baseline on the x-axis against firing rate after deletion on the y-axis. And everything is clearly shifted up to faster rates. And while this does have the effect of driving a supercritical regime, it's challenging to disentangle that from the basic effects on firing rate. However, within this model, we can make much more subtle manipulations. Let's just go back to the baseline model again here. And this time I'm going to delete layer 2, 3 somatostatin cells. This is the top yellow band. And while there is going to be a very slight reorganization of the network, it's quite difficult to catch. So let's try this again. So I'm going to count down 3, 2, 1. There. I think you really have to squint to catch that. But if you look at the firing rates, you can see that we have certainly not destabilized the model. There's really no change in the total number of spikes. But we do see that this changes DCC and some of the information encoded within the network, giving us a better, plausible biological mechanism for emergent properties of the network. So ultimately, this class of model will lead us to targets that we can manipulate in freely behaving animals so that we can start to understand how these emergent phenomena, such as criticality, contribute to behavior and cognition where it really counts. And with that, let me wrap up and give full credit to my laboratory, the people who fund the science that we do, and my spectacular collaborators, especially Ralph Wessel and Zheng Ma, who drove the majority of the paper that I shared with you today. So, thank you very much again.
Thanks a lot, Keith.
Anytime.
Yeah, so yeah, there's always some questions here already. Well, from Joshua Welsh. Keith, thanks for the fascinating talk. Do you know whether artificial neural networks, for example, those used in machine learning, deep learning, also show criticality?
That is a wonderful question. And there has been, I'm going to quickly step outside my realm of expertise. So please forgive me if I put my foot in my mouth, but I know that there has been some research to suggest that some of these deep learning networks that can be tuned to criticality gain some powerful computational properties. And some of the neuromorphic chips have also begun to sort of include this as a parameter and helping to sort of tune them to a computational optimum. But I don't know what the sort of current state of the art is in that field. But I think that those folks are well aware of this and that it is not irrelevant to them.
And then there's one from Hyeyong Shin. Keith, super interesting talk. Can you comment on the debate on whether criticality explains the 1/f feature of LFP power?
Oh man, that is a deep and long debate. I don't know if we have time to separate that right here. I think that that's going to take some careful experimentation. But that's a good question.
Okay, then also one from Shinya Ito. Keith, the slide of the time courses of firing rate and DCC looks like the neural network even prioritized fixing the criticality, and the firing rate change is induced as a consequence of it, which is then later fixed. Is the criticality and overall firing rate linked? Is it through a specific approach by the network to keep criticality of the network?
So that is a fantastic question, and you're very clearly paying close attention. So thank you. Let me just say first that this is a speculative answer, but my guess is that the critical regime of the network is probably driven by the inhibitory firing rate change. So if you look back at a paper we published in 2013 in Neuron, the inhibitory neurons do collapse on the first day of mononuclear deprivation. And so everything that I showed in slides here is from RSUs. That's the presumptive excitatory population within the recording. So I think that the inhibitory neurons are imposing this computational regime on the excitatory neurons. And I mean, speculatively guess that the firing rate stabilization is a sort of a longer term response, while the quick reorganization of the sort of emergent properties has to do more with the fact that the animal isn't rendered useless as soon as the eye is sewn shut. So there have to be some more rapid mechanisms, but the timescales on which these play out is complicated, and it's totally unclear how they interact with each other. And that's where I hope that models like the one from the Allen will let us begin to sort of twist some of those knobs and understand perhaps how cell level changes or synaptic specific changes actually feed back on these higher order regimes. But it all points back to inhibition.
So there's one more from Shan Kim. What's the relationship between criticality and EI balance? From your model, what's the self-organizing mechanism to enable the network poised at the critical point?
I don't know if I can point to the mechanism yet, but in critical networks, kind of by definition, have to have EI balance, right? So that's why I was saying that the criticality basically represents a optimizing all of the features you can basically think of for a network sort of simultaneously. So you have to have gain control, you have to have EI balance. This also then maximizes the sort of total entropy within the network, and sort of simultaneously arrives at this mythical state. But EI balance does fall out of this regime.
I also want myself is, I mean, if these avalanches that you mentioned is, if you just take the original, like the Allen model, do they show this avalanche behavior?
They do. They definitely do. So in the baseline model that we got from Anton, when we run the point process model, because it's much faster than the fully biphysically realistic one, the system is actually very surprisingly, it's close to criticality. Its DCC I think was about 0.3, which is not within the limit of what we call a near critical system in my laboratory, but it's certainly not as destabilized as the early hours of mononuclear deprivation. So, and if you think back to that three-dimensional plot I showed where we had a very small and discrete solution space, basically suggests that as Anton and his team, or Gaut, as you guys were picking from a range of parameters that were published, you got pretty close to a viable solution. And so we've been able to tune that model a bit, sort of search another parameter space, and very easily get it to generate critical dynamics. But the avalanches are a feature of that model right out of the box.
Here's one from Alexis Ducote. Keith, given experimentally observed alterations in EI balance with aging, might we expect deviations from criticality to explain some of the deficits in sensory acuity, encoding, processing speed observed in normal aging or age-related pathologies?
Yes, that is a fantastic question. So, I mean, the tempting leap to make here, and one that I have some students in my lab that are pursuing these types of questions, is that it is ultimately this higher order regime in the cortex that sort of allows it to do the things that it does. And so anytime that you have pathology or sort of an unraveling of those functional processes, it should be reflected at the level of the sort of ongoing computational regime. And we do see some evidence for that already in certain models of aging or pathology. And so then the bigger question is, can we now go in, identify mechanisms, and actually restore criticality and see if it reverses the cognitive or behavioral impacts of pathology and aging? But yes, that is a very powerful and important link that needs to be addressed.
The next one from Anton. What do we know about criticality in other parts of the brain, say outside of the cortex?
So let me just sort of show off my lab for a moment. That my team has developed the ability to record from up to 12 brain regions simultaneously. For longest recording we've run is 15 months. And so we collect up to almost 1,000 neurons that we can track continuously for the entire time. So we're talking sort of petabyte scale data sets. And when they do this, we can then really begin to explore whether or not we have regional differences in criticality, which we do see, and whether or not we see intra-regional criticality, which we also see. But we've seen criticality in the striatum, which has some interesting properties, in superior colliculus, throughout the hippocampus, and sort of all over the cortex. I think the striatum is probably the most variable region that we've looked at, but those are preliminary data, and I wouldn't be willing to kind of, hang my reputation on that yet.
And there's one from Upi. Could you not imagine sort of like signaling feedback, say through astrocytes to achieve criticality?
Yes. I think that this question, if you zoom out a little bit, is kind of the essence of what makes emergent phenomena very hard to study, is that this is a dynamical system. Think about a car. You're not going to be able to reduce driving to a spark plug. So all of the components matter. And so I think the larger question is, are there levers that you push that have kind of maybe an amplified control over this effect? I mean, I'm sure that pushing on astrocytes, pushing on microglia, pushing on all of the different classes of inhibitory neurons will have some effect. Some of them, however, might have sort of an overrepresented effect. And that's where we want to begin to go in and really start to tinker and play.
There's one more, and one from Marta Gajova. Keith, can the criticality of the system depend directly on like connection strengths between subtypes of cells?
I don't know. That's a very good question. I'm sure it can. I guess I'm sort of open to all of this. And I guess what happens in my mind is that as any aspect of the system is tuned, there must be kind of compensatory and immediate feedback. I mean, this really points to the work of folks like Yves Marder, who really show these sort of degenerate solutions to arrive at a stable system, but just now expanded to the cortex.
Okay. And then the final one is also from Alexis Ducot. Also, Keith, in your experiments, do you observe any of the state-dependent, say like attentional sleep, for example, fluctuations between sub and supercritical states that may allow for dynamic shifts between regimes, or evidence for the weakly correlated non-avalanche criticality found in Dahmen et al. 2019?
I would love to answer that question, but I don't want to scoop my own student. And so I'll just say, remember that, and hopefully you'll see something in the literature in the next year. But let that be a hint.
Okay.
All right.
With that hint, we move on. Well, thanks again.
Thank you very, very much.